So the next stage after prophase is the metaphase, which is the middle stage. Middle stage. So the former speaker said in the prophase, cells are being condensed. So the condensed cells align in the equidistance of this cell, and the imaginary equidistance is known as the metaphase plate. So in this stage, three processes occurs. The first stage is the cell alignment, that is, cells align in the metaphase plate of the cells. The next process is the attachment of spindles. So these are the spindles, or you can also call them centrioles. They attach themselves to the chromosomes, and the chromosome consists of two sister chromatids. So the next process and the last process is tension generation. This is not really considered as a process, because once the spindle and the centrioles are attached to the chromosome in the metaphase plate, they intend to create a tension between them. So the next phase after the metaphase will be able to occur, that is the anaphase.
Now the anaphase, that is the fourth phase. This is where the sister chromatids begins to separate and move to the poles. First, you can see here that they are the same, both red, both blue. But yeah, they will first separate themselves. After separating, they will begin to move to different poles. When they are moving, this is caused by the elongation of the spindles, like you said here, that is connecting them. When they elongate and move towards different opposite sides, they will separate, and then the telophase will begin.
After the anaphase comes the telophase, and telophase is formed when the chromosomes, they cluster into a differentiated body. They cluster into a indefinite body, in which the daughter cells are ready to separate, and at that form the nucleus begins to dissolve, and then the ER begins to form, in which the cell walls begin to solidify, and then the two nucleuses begin to separate, and after they have both separated completely, the telophase is complete.
I'm here to describe meiosis 1. As it is said in As was said before me, there are two cell divisions, which is the mitosis and meiosis. I'm here to discuss the one under the second one, which is the meiosis. Meiosis is a special type of cell division in which a single cell divides in which a single cell undergoes two rounds of cell division to form four haploid cells. In meiosis ensures genetic diversity. The first step in meiosis is the phase 1, which the next speaker after me is going to come and describe. But in meiosis, it ensures genetic diversity in the cell, and for meiosis to occur, there must be the exchange of nuclei or gametes between two parent cells, which is the male and the female. For example, I said it ensures genetic diversity. For example, in spirogyra undergoes two types of cell division, but using the meiosis own, they make sure that it makes them, for example, if they are in a place where there is adverse condition, it will make them like the offspring of the spirogyra. It will make them to be able to adapt to the condition, to the new environment.
Phase 1 is the first stage of the meiosis cell division. It has three stages. These are the early prophase stage, the mid prophase stage, and the late prophase stage. In the early prophase stage, the chromosomes become visible as long threads. In the mid prophase stage, each chromosome is paired with their homologous partners. These are matching partners. So they are seen as these red and blue matching partners in the cells. In the late prophase stage, the chromosomes, each chromosome produces a double-stranded replica of itself. Then the nuclear membrane of each cell starts disappearing. So it's in this stage that cross-links called chiasmata are produced. These chiasmata help to hold each matching chromosome together. And it's also in this stage that each matching chromosomes are formed because of variation and mutation.
Okay, in this stage of meiosis, which is metaphase, the M stands for